Hello friends, welcome back to my YouTube channel. Today in this video we'll be going to talk about the reporter genes. So in previous topics we have discussed about the yeast to hybrid system and phage display, which is a type of protein-protein interaction, and in this video we'll be going to see about the reporter gene. Okay, so the reporter gene is nothing but it is a gene producing protein. Okay, reporter gene can be defined as gene producing protein which can detect or quantify a simple assay, and it can detect the colors. Okay, so if we see the most commonly used the reporter genes are laxid, which is also called as beta gal, luciferase, GFP, and cat. So actually these three are in your syllabus, so we are going to learn about these three reporter genes. Okay, what this reporter genes do? Actually the promoter, enhancer, and transcriptional factors, these are all the part of transcription. Everyone knows that if you have studied transcription in your previous semesters, you might know and familiar with the terms like promoter, enhancer, and transcriptional factor. These factors binds with the reporter gene and make a use of reporter gene to produce a desired protein. Okay, to produce a protein. Now we'll be going to discuss about the luciferase reporter gene. Okay, what is this luciferase? Actually this luciferase is present in a firefly. Firefly is a jugnu, you know that. In our terms we called it as jugnu. So it will produce a bioluminescence. Bioluminescence means nothing but without the external illumination it will produce the light. Okay, that we called as bioluminescence. So luciferase example is firefly. It is seen in firefly, and it used in the study of gene regulation and biological function of it. Okay, and if we talk about the light it produced is yellow green light it will produce. Okay, and the equation for this luciferase is luciferin in presence of ATP and oxygen, it will gives or it will generate oxyluciferin plus light. What light it can produce? Yellow green light. Okay, so this is the equation which I haven't mentioned in my notes. I didn't mention in my notes. I will mention it on a side, so you can write this is important equation for this luciferin. Okay, and now if we talk about the diagram of this luciferin, the promoter here, promoter, and here RNA polymerase. Here the transcriptional factors will be attached. These are the transcription factors, and to this promoter our luciferase and enzyme will be there. This is our reporter gene, reporter gene will be attached. What happens upon transcription? Upon transcription it will produce mRNA, and upon translation it will produce enzyme. What enzyme? Luciferase enzyme. And this is, we called it as reporter protein. Okay, and this enzyme with the attachment of substrate, it will form a light. Okay, it will form a light, and this light sequence we called it as light signal is equals to light expression, and light expression will also be equal to promoter activity. Okay, that you can say. This is about the luciferase. The equation is important and this diagram is important, and you need to write some point about the luciferase. That's it. Now we'll be going to see about the other, that is beta gal. Beta gal is also known as LAG-Z. Okay, and it is encoded by lag gene. Beta gal is encoded by a lag-Z gene, and it is derived from E. coli. It is derived from the E. coli. What it will do, it hydrolyzes the beta gal. It hydrolyzes the it hydrolyzes, or it catalyzes the beta gal, sorry, it will catalyze the beta galactosidase to hydrolyze X gal. ओके, टू हाइड्रोलाइज एक्स गैल। ये बीटा गैलेक्टोसिडेज को कैटालाइज करवाकर हाइड्रोलाइज करवाता है किसका, एक्स गैल का। तो आपको पता है लैक्टोज क्या है, लैक स्टैंड फॉर लैक्टोज। लैक्टोज जो है हमें क्या देगा, बीटा गैलेक्टोसिडेज एंजाइम के प्रेजेंस में जो है ये हमें देगा ग्लूकोज प्लस गैलेक्टोज, ठीक है, ग्लूकोज प्लस गैलेक्टोज। और एक्स गैल का जब हाइड्रोलाइज होता है इन प्रेजेंस ऑफ दिस एंजाइम बीटा गैलेक्टोसिडेज, ये एक्स गैल क्या है हमारा, कलरलेस है, कलरलेस है। तो ये क्या देगा हमें, व्हाट इट विल गिव अ गैलेक्टोज, गैलेक्टोज प्लस 4 क्लोरीन 3 ब्रोमीन इंडॉक्सी। ये क्या होगा, ये हमारा जो होगा डीप पर्पल कलर, डीप ब्लू कलर में होगा, सॉरी, दिस विल बी इन अ डीप ब्लू कलर। तो ये जो ब्लू कलर है ये डराइव होता है हमें किससे, बीटा गैलेक्टोसिडेज से। सो व्हाट इज ल्यूसीफेरेज इज, ल्यूसीफेरेज जो है एंजाइम बेस्ड रिपोर्टर जीन है, ठीक है, इट इज़ एंजाइम बेस्ड रिपोर्टर जीन। Luciferase is enzyme based reporter gene, and beta gal is calorimetric based reporter gene है, ठीक है, बीटा गैल इज कैलोरीमेट्रिक बेस्ड रिपोर्टर जीन, ठीक है। एंड इफ वी टॉक अबाउट द GFP, what is this GFP? GFP is stand for green fluorescence protein. It is stand for green fluorescent protein, and it is also known as GVGFP. Why GVGFP? AV stands for Echoria victoria. It is a jellyfish, and GFP was first isolated from this jellyfish Echoria victoria. That's why it is also known as AVGFP reporter gene, okay. And it is an autofluorescent protein. Auto means self fluorescized protein it is, and it has 238 amino acid residues. That's why it has variants. GFP has variants, okay. Why it has variant? Because it has 238 amino acid residues in it, and it produces a different color of lights for a different amino acid, okay. And this GFP is also used as fusion tag protein, okay. GFP is used as fusion tag protein, okay. And the variants of it is yellow. This is based on the mutation of threonine at place of 392 tyrosine. If threonine is changed to tyrosine at 203, then the color will be a yellow. And other one is blue fluorescent protein, that is modification at place of tyrosine 66 to histidine. Tyrosine 66 to histidine, it will give a blue color. And cyan variant fluorescent protein, tyrosine 66 position changes to tryptophan, then it will give us cyan fluorescent, okay. So what GFP will do when it is acting as a fusion tag protein? When it acts as a fusion tag protein, it will monitor the protein localization मॉनिटर करेगा, और इसके लाइट प्रोड्यूस करने की वजह से डिफरेंट काइंड ऑफ लाइट्स जो प्रोड्यूस करता है डिफरेंट वेवलेंथ पे, दैट्स वाई इट इज़ कॉल्ड एज यूनिक रिपोर्टर, okay, that's why it is called as unique reporter. And this GFP is fluorescent based reporter gene, ठीक है, fluorescent based रिपोर्टर जीन। तो हमने देख लिया रिपोर्टर जीन क्या है, जीन प्रोड्यूसिंग प्रोटीन है और ये कलर्स को डिटेक्ट करता है। और फिर हम ये भी देख सके कॉमन वाले कौन से हैं, फिर ल्यूसिफरीन के बारे से। देन वी हैव सीन अबाउट द ल्यूसिफरेस detail, then again beta gal, then later GFP. This is how we are going to end this topic, and if you are new on my channel, please subscribe my channel, share my channel with your friends. Thanks for watching.